All right, thanks Karen, thanks Kat for the invitation. So if you like this symposium, Kat and I were preparing to have like a second day next year. So this is going to be merging the stem cell program in the clinical center. So it's going to be even more exciting. So my lab focuses on stem cells for the use of modeling diseases in a dish. And most of the time we focus on diseases such as autism or disorders that affect the social brain. Most of the time those disorders, they start in uterus. And that's one of the major bottlenecks for the development of new treatments and therapies for these conditions, since the access to the material is quite limited, especially at very early stages in neurodevelopment. So we have to rely on non-invasive techniques that don't have the high resolution that's required for experimental biologists. So instead, we use stem cells models, and you probably heard about the mini brain a dish or brain organoids. And I think most of the time, the media kind of exaggerate on how these cells look like, and I wish they could look like that, but they do not. So right up front, I will state some of the limitations of these technologies so we are all on the same page. So these are brain organoids that are composed by vastly mature neurons. They are not vascularized. They do not contain all the cell types that you would find in a normal brain during development. For example, they do not contain microglia or the endothelial cells that would make the blood vessels. Also important, we don't have the ideal culture, so we don't know how the brains develop in uterus, so we try to mimic using empiric methods and medias and factors. Most likely we're getting it wrong. So for all of that, I mean, it's a model and has its own limitation. One of the important questions that I think our contribution is moving towards is this idea that can this technology be translational, meaning that what you see in a dish, can you also relate back into the brain of the patient? So for the brain, this is a little bit difficult. So in the past years, we have been working on a new method to generate these brain organoids, and we start from pluripotent stem cells. This could be either human embryonic stem cells or iPSCs. So cells would be dissociated into single cells, and then you start the induction phase. So the cells will neuralize. You can drive them to different brain regions depending on the factors that you add to this culture. And after that, you add EGF and FGF at certain time points just to stimulate these neuroprogenitor cells to proliferate. After that, you remove the growth factors and let the cells do what they do by themselves. They will self-organize, they will migrate, and they will form these more complex structures, and they will mature. And we can keep these cells or these brain organoids in the lab for several months, if not years. And for those who have never seen them, this is Isabel, a postdoc in the lab, just handling a plate full of those brain organoids. I hope you can see all these white dots in there. Each one of them is one brain organoid. So two things here to stress out. They can reach about 0.5 centimeter, so they get that big. You can actually see at naked eye. And this is one batch of differentiation. So when you induce the cells to differentiate, you don't get one or two brain organoids. You get several, if not thousands of them. And this has several advantages. You can use the same batch for different experiments. You can do RNA expression, electrophysiology, and so on. So this is all good. So the bioengineer part of this talk is actually done by the cells itself. So they do what they do in uterus, and they are programmed to migrate from a proliferative zone. So this is just a representation in 2D of what the cells will do in 3D. Progenitor cells would migrate, and they follow up cables. These are cables provided by radioglia cells, where these progenitor cells can use to guide them and migrate towards the outer part of the organoid. As they reach out to this outer part, the cells will stop migrating, start the differentiation process, and these neurons will make connections to each other, forming the cortical plate. So in a cross section of these organoids. This is a typical picture. This is a 4-month-old organoid. So you have a ventricular zone full of progenitor cells surrounding it, and as the cells migrate, they form the cortical plate, and those neurons slowly would form the connections between the different layers. So as you mature, the definition between those layers in the cortical plate becomes clear. So what are the cells that we have in there? So we have been looking into that in collaboration with GeneEO here at the Consortium, doing like single cell transcriptome analysis, and this is just like a snapshot. And I think Sally mentioned in her talk yesterday how important it is to do the analysis in a dynamic way, and I always stress that in a moment. So at 4 months, the vast majority of cells that we have are glutamatergic excitatory neurons. You start to see a definition between these two populations. These will become the upper and lower cortical neurons. There is a population of GABAergic neurons that are very discrete but will increase over time. The progenitor cells also will decrease over time in the amount of glia cell. Mostly astrocytes will also pick up between 4 and 6 months. So how do they compare to the real brain? We use tools such as the brain span, which is a collection of gene expression coming from postmortem tissues, and we compare to our iPSCs and organoids at different stages. And there are other tools such as the cone test that you can use to actually match brain regions in developmental stages. So these are different iPS cells, different clones that we have. At the iPS cell stage, they all match pretty much embryonic cells. 1 month organoid, you start to see that the matching is going to be more into the early fetal cells, and a 3-month-old organoid is already matching like a late fetal stage during development. So this is by gene expression. Of course, gene expression has lots of limitations. The postmortem tissues that you get are not ideal, and there are RNA degradation. So we do the best as we can with this model. And in the past, that's what my lab and other labs have been exploring. We're exploring the structural formation of these organoids and the gene expression of these organoids to understand a little bit of neurological disorders or conditions that affect brain structure. And perhaps one great example is the Zika virus and microcephaly. So we use these two to really show causation between the Brazilian circulating virus and the killing of the progenitor cells that will give rise to a cortical plate. So in the presence of the Zika virus, we see that there is a deficiency in several populations of cortical neurons that will lead to this macrocephalic phenotype. The other thing that it's important, since we have plenty of these organoids, we could use them to do drug screening, and that's in a low throughput, at least for now. And that's how we came out with this drug called sofosbuvir. That's an FDA-approved drug that can cross the blood-brain membrane and can also cross the placenta to reach out to the fetus. So not only the drug could inhibit the replication of the virus, but we could protect, at least in an experimental model, pregnant animals that are infected with Zika. So we block vertical transmission. The fetus are born without the virus. So this is all good news, and it's a fast way to repurpose drugs, and at least for this experimental condition. So, but I mean, we and others, we are always thinking that, well, I mean, but the brain organoids, we need to have any evidence that they do have network formation. And for a while, I mean, we were kind of discouraged that they would kind of reach to the level where we could detect some kind of network in there. And so we decided that the best way to measure the networks would be to plate these organoids on top of multi-electrode arrays. So we have plates like these where the electrodes are printed in the bottom of the dishes, and you just plate the organoid on top of them, and these electrodes will capture the electrical activity that's passing by, generating activity maps like that, or raster plots describing the activity of each independent electrode or channel here. And so just for us to have like a common nomenclature here, every trace that you are seeing here is one spike event. This is a field electrophysiology, so you cannot claim that's coming from a single neuron. So it's just an event that you are capturing, passing through that electrode. Lots of events at a very short period of time, we call that a burst. And if you have bursts among 5 or more electrodes, it means that they are probably connected, so we call that a synchrony event. So the beauty of that is that these organoids remains alive. You don't destroy the tissue, you don't destroy the organoid. You can keep them in the incubator and keep recording them over time. So you can follow up how the networks really evolved. And in the beginning I said that we were not like having some high expectations for that. And this is because the data coming from the literature was not super excited. So I'm plotting here just for you to have an idea, the mean firing rate in hertz, which is overall brain activity that you can capture using these multi-electrode arrays. And what we did was a meta-analysis on all the works out there, both in mouse or rat primary cultures, as well as in iPS-derived neurons, to look for papers describing the use of this technology. And as you can see, I'm just listing a few of them. Most of these works are keeping the activity below 5 hertz. It's actually really hard to have a 5 hertz activity with human iPS-derived neurons. So where do you want to get? We want to get to something close to the 20 hertz. So for you to have an idea, the mouse, a slice of a mouse cortex would be around 18 hertz. And people have been sticking these electrodes in primate brains, and they are reaching something close to 20, 21 hertz, which probably would be similar to the human brain as well. So with the organoids, we have the advantage of longitudinal analysis, and people have tried to actually measure the network activity over time. So this is the work from a colleague, Paola Lotta in Harvard, and she was able to keep the organoid for 16 weeks and 32 weeks with electrodes in there. But over the course of all these weeks, we actually don't see an increase in network analysis. So what I'm going to show now, it is weekly measurements of these multi-electrode arrays using this protocol that I just described. So each point here is an average of 8 organoids, again, every week. And you can see that by 16 weeks, we already have like something that's similar to a human iPS-derived neurons in a 2D system. But I think the biggest surprise was when we keep the cultures for that long period of time, and we see that the electrical activity really picks up in about 6 months, and it reaches something close to the 20 hertz. So if I translate that into numbers, you can see that the number of spikes per minute, it will move from something that is around 3,000, 5,000 spikes, something that we do see in 2D cultures, to something close to 300,000 spikes, never ever described in tissue culture before. So we are reaching a level of activity that's quite high. As a consequence, the interval between those bursts are reducing close to 0 here, or to 1, meaning that all these neurons are highly connected among themselves. I also mentioned that in this unique system, we have both glutamatergic and GABAergic neurons, and we perform some electrophysiology just to make sure that all the networks pieces are working as we expected. So I won't describe these in details, but just to say that if you treat with, for example, bicuculin to block GABAergic receptors, you go on and increase your activity, or if you block with mucinol or TTX, you eliminate the activity from these brain organoids. You can wash it out, and the activity will come back eventually. So if we have that much activity, it means that we might be having other kind of activity in the brain, such as brain oscillations. And this is not your typical 2D network oscillations that you can measure in the culture. We are talking about gamma, alpha, delta oscillations that you can capture, for example, using EEG. You can only record from EEG these waves because there's so much activity in the brain, it passes through the skull, the electrodes can pick it up. So we team up with Brad Wojtek, a cognitive neuroscience professor here at UCSD, and he helped us on this analysis. So what we did was we went back to our data on the MEAs, and we use filters to generate LFP, and these would be the data that we use to confirm or validate these brain waves. One thing that was quite striking is that in the beginning we see no oscillations, but the oscillations start to appear between 4 and 6 months, and they are quite irregular in the beginning, something that would mimic a pathological condition or early stages of development, and again they become irregular or more complex over time. And this is there is a nice correlation here with the activity or number of spikes that you can get from these brain organoids. So another way to show the waves is actually by plotting the population spikes or the voltage here. As I mentioned, by 2 months, there is no much activity. 4 months, we start to see some peaks. 6 months, it's pretty clear that you have oscillations that are stereotyped. And by 8 months and longer, they become completely irregular, as you would expect for something that's complex. So because of this complexity, if you just plot the number of peaks over time, what you have is this inverted U shape, and this is caused by a rebounding effect of these oscillatory waves. So the peaks start to overlap again. And the only way for this to happen is because you're moving from a system that's quite static to a more flexible system, such as described here in this cartoon. So what we are having is really intra-connections between the different subtype of neurons in these organoids being captured, at least on the superficial part from the multi-electrode array. So the next question that we had was, well, I mean, if you're seeing or detecting these waves, maybe we can compare them to the human brain. And so to the human brain, as I mentioned, we can do that using EEGs. And an adult EEG would look like that. And our organoid EEG looks like that. So there are parts where the organoids are quite silent. We don't see these waves, but eventually you can see this transient activity followed by another period of quiescent time where the brain is just silent. This is exactly what happens in preterm babies that had EEGs in there. So it looks like we are mimicking very early stages of development and not really the adult human brain, which is in a way kind of expected for the system that we are having here. So what we decided to do was to create an algorithm, a training machine, where we plot a little bit over 500 EEGs from preterm babies born with 25 and 38 weeks. And we trained the machine to recognize the pattern of these EEGs and to give and to guess the correct age of the individual. So the machine can do that really well. We trained the machine, and once the machine was calibrated, we start feeding them with our data coming from the organoids, hoping that we'll predict something close to the human brain. And so the data that we have follows like that. The perfect prediction would be here on the red bar, and our prediction with all the time points is in the black curve here. And I hope you can appreciate that the best predictions, or when the machine kind of gets confused, it happens after 25 weeks. So after 25 weeks, the prediction is actually really good. Below 25 weeks, we see a really bad prediction. And the reason is that we could never train the machine with EEG below 25 weeks. The human brain just cannot survive. It is possible that in the future, with new technologies, we will have access to EEGs of the human brain below 25 weeks. But right now, that's what we can do. But again, it was a really good surprise to see that there is a correlation between the organoid development in the human brain based on these EEG features. And I'll give you just one example here. And this is what we are measuring here is what I told you, the distance between these sporadic or activity. So it's the quiescent time that as you grow older, the quiescent time in the brain becomes shorter and shorter. And this is what happens with the organoids over time. The quiescent time becomes shorter and shorter. And by 40 weeks or 9 months, it matches the 9-month or a postnatal human brain. So this is not the entire EEG, it's just some features of the EEG. But nonetheless, I mean, it suggests that we are following some trajectory that surprisingly mimics the human brain. This, again, was not what we would expect from just balls of cells in a dish. So finally, one interesting thing is and again, I mean, in our discussions with the Wojtek lab, we were excited that we could see these oscillations. And one thing that he pointed to me, he said, you know, you now have an experimental model to understand how these oscillations appear. Because so far, all the cognitive neurosciences, they have been doing comparative work. What happens in a determinate state, for example, in a disease state or awake or sleeping state, and the correlation of these brain waves. But nobody knew how these brain waves actually emerge from human development. We could never measure that. So the experimental model that we decided to try was, well, I mean, we could use drugs, but we could use also genetics. And we have been creating organoids that lacks specific genes using CRISPR. So these are full knockouts on genes that are related to neurodevelopment. And one gene that we like a lot is MECP2. If you don't have MECP2, most likely you're going to die prematurely. If you have just one copy of the functional gene, you're going to have Rett syndrome. If you have less dramatic mutations in this gene, you're going to have autism or schizophrenia. So the full knockout gives virtually no brain waves. Even though you do have some activity on these neural networks, the oscillations will never emerge. And so we try to understand better how come a single gene can abolish the emergence of these oscillations. So again, we turn into single-cell analysis. We dissociate isogenic organoids, the knockouts and the controls, and we perform some gene expression to look for specific cell populations that might be affected by this gene at these early stages. And what we find out is that layer 5 and 6 has a significant reduction. If you zoom in that specific population and ask what kind of neurons are differently expressed between the mutants and the controls, we get two populations, the CTP2 positive neurons and the STAP-B2 positive neurons. These are two populations that we see that are reducing the knockouts compared to controls. So it might be that this is the reason why you don't have the emergence, or at least the spontaneous emergences of these oscillations. You probably need the right amount of neurons in each populations to reach out to a level where we can see the spontaneous generation of brain oscillations. All right, so I will stop here with my take-home message. So I think I show you, and I hope you appreciate that we can generate spontaneous network, dynamic network over time. And these networks coming from these brain organoids, they follow the biological program. So this is something where the environment is controlled. So it's all coming from the genetics. It's pre-programmed to do exactly what it's doing here. We see these delta high gamma oscillations that are coupled with the number of spikes, or probably the synapses. And MECP2 is one of the genes that we see that can mess up with these cortical oscillations. And it's consistent with clinical phenotypes of people who are born without the gene. I mean, there's the EEG from those patients is all messed up, and they actually barely survive. And if we compare the organoid electrophysiology with the preterm babies, we see that mostly they mimics the 28 to 38 post-conception weeks. So this is, I think, our first step towards creating a model that is more translational. I'll finish here by thanking people in my lab, the families that participate with us. As I mentioned, all the oscillation analysis was done in collaboration with the Wojtek lab. Jean Yu helped us on the single cell analysis. Lilia lab helped us on some of the gene expression. This work was largely funded by NIMH and CERN. And I also have to disclosure that these generated a startup company, and I'm a co-founder of Tismo. It's a company that uses these brain organoids for drug discovery for autism. I finish here, and I'm happy to take any comments and questions. Thank you.
So I assume your knockout was a male for MECP2. The MECP2 gene is on the X chromosome. Have you looked at females, the heterozygous state?
Yeah, that's a great question. We have not. So far, we just work with the knockouts. We thought that the knockouts would give us the most dramatic effect, and indeed it does. So to create a mosaic, we'll have to mix cells between the wild type and the mutant clones and mimic something that would happen in a female brain. We are doing that right now. Yeah.
Alysson. So that was fascinating. So I'm wondering, so for the last part, can you just take the wild type organoids, dissociate, and then sort out the different neuronal cell types, and then spike them back into organoids at different proportions, and able to figure out what's the right mix that would lead to the oscillation?
So that's a great point. We've done something similar, and I'll tell you why this experiment will not work for the cell populations. We were able to get one organoid, dissociate them, and put them back. So they have the same number of neurons, they have the same subtype of neurons, and they aggregate in 3D. They will never show oscillatory waves again. So you need a developmental time, the microcircuitries to be formed inside the organoids to really have these oscillatory waves. By just putting the same number of neurons and subtype of neurons in there, we don't get it. So we thought about these experiments, but because of the data, I don't think it's going to be informative.
Thank you. So beautiful. So I have a question about the difference between your approach and Paula's approach. Can you say anything about the key factors that lead to the improved maturation of the organoids with your system?
If I'm not wrong, what Paola uses was the protocol from Jürgen, where you don't drive the cells to specific brain regions. So these are embedded matrigel organoids, and they will develop different brain regions at random. So it could be that where the electrodes were inserted would not have that much activity. It could be many things. We've never done like side-to-side comparisons to really claim that there is something specific to the protocol. I think that if you drive them to a specific brain region, there is higher chances for you to get something more organized.
Great. And one last question. Are you using optogenetics to drive activity of different neuronal subtypes?
We are, but all the data I show you, it's all spontaneous. There is no induction.
Yeah, I know, but I figured it would be interesting to see how that alters the oscillatory profile.
It's coming. Right. It's coming, Sally. Thank you.